Good afternoon everyone. So today we are going to see about biochemical characterization of bacteria. I am Arunadevi from microbiology department, and today we are going to see about the bacterial characterization using biochemical. So what is mean by a biochemical? So biochemical is nothing but a metabolic derivative of a living cell. Okay, each and every cell will be doing the metabolism. So from that metabolism they will be getting a energy products. So that product is said to be the biochemicals. So with that biochemicals, they will be doing all their energy activities as well as their reproductive activities, and every mechanism will be done with the help of the biochemical substances. And so today we are going to see about how we can able to characterize an organism using their biochemical properties. So the common biochemical characterization we are going to see today. So the first test we are going to see is the indole test. So indole is a product which has been produced from the amino acid called tryptophan. So this tryptophan will be present in the peptone water. Okay. So when the organism decomposes the tryptophan, so they will be converting the tryptophan into indole. Okay. So this enzyme needed for the tryptophan conversion is the tryptophanase enzyme. Okay. So the medium contains water and the tryptophan. When the organism produces the tryptophanase enzyme, they will be converting the tryptophan into indole and the pyruvate and the ammonium products. Okay, so with this we can able to confirm the production of indole by adding a reagent called Kovacs reagent. So this Kovacs reagent, when it is added to the medium, they will be converting the medium color, and then we can able to detect by the presence of pink color ring formation on the top of the medium. So how will you do this test? How will you perform this indole test? So just by inoculating the organism into the peptone water, after that we have to incubate the organism at 37 degrees for 24 hours. So after that, by adding the Kovacs reagent, we can able to see two reactions. So if the medium contains the indole, okay, so for example, E. coli is an organism from Enterobacteriaceae family, so they can able to produce a positive reaction. So that positive reaction will be seen like this. So they will be producing a cherry red color ring at the top of the medium. So this is the positive reaction. And negative, so certain organisms like Klebsiella, so it is also in Enterobacteriaceae family, but they are negative for the indole production. Okay, so they will be producing yellow color or green color, any other type of ring will be produced in case of negative test. Okay, so this is the first biochemical test we can use. Next is a methyl red test. So methyl red is a common test used for the detection of mixed acid production. Okay, so each and every organism will be undergoing the fermentation process. So they will be converting the glucose molecule into many different varieties of acids and alcohols with the liberation of acid. With the liberation of acid as well as the carbon dioxide as the end products. So this can also be detected by a special type of test called methyl red test. So this methyl red test, in that test, the organism will be metabolizing the pyruvic acid, the end product pyruvic acid, by mixed acid pathway. So this mixed acid contains lactic acid, acetic acid and many other acids. So this acids will be maintaining the acidic environment in the medium. So that acidic environment can be detected by adding the methyl red reagent. So that methyl red reagent will be detecting the low pH. Okay. So the pH will be 4.4. So when it is 4.4 means it will be low pH. So in that pH, the methyl red will be converted the medium into red color. So this denotes the positive reaction. So this is the principle of methyl red test. Okay. So what is the medium used for the methyl red test? So the medium used is the MRVP broth. So what it contains? It contains buffered peptone, dextrose, dipotassium sulfate. So we have to maintain the pH of 7. So same procedure. So for the liquid medium we have to go for inoculation with the help of sterile loop. So we will be inoculating the medium and the we have been inoculating the pure culture or any other test organism we are going to see, and that should be inoculated in the MRVP broth, and after incubation of 37 degrees for 24 hours, and then with the help of methyl red reagent. Okay, so we have to add the methyl red reagent up to 5 drops. 5 drops we can able to detect the test reaction. So if it is positive means it will be turned into red color, and it is negative means it will be turned into yellow or orange color. Okay, so this is the detection of low acidic pH, and this is a high pH. Okay, so if the mixed acid production has been done means here the positive reaction, red color will be formed. If there is no acid or less higher number of acid production means that will be negative in the. So the next test we are going to see is the August-Proskus test. This is the test, same mechanism. So in case of methyl red and then August-Proskus, the same medium has been used, but certain organisms they will be undergoing the alternative pathway for their metabolic activity. So in that alternative metabolic pathway, they will be producing a compound called acetyl-methylcarbinol. Acetyl-methylcarbinol. So that acetyl-methylcarbinol can be detected by using this August-Proskus reaction. Okay, so this is the specific principle behind the VP reagent. So same mechanism, after the metabolic activity, they will be producing same acid and alcohol. Here, a special derivative called acetyl-methylcarbinol will be synthesized by the organism, so that glucose to acetyl-methylcarbinol can be detected by the August-Proskus reaction. Okay, so what is the procedure means? Same procedure. With the help of sterile loop, we will be inoculating the test organism or the pure culture into the medium. After 24 to 48 hours, we will be adding the reagent called August-Proskus reagent, VP reagent 1 and 2, and then we can able to see the reaction. So the eosin pink colored, that medium will be turned into eosin pink color, so that detects the positive reaction. If it is yellow or orange color means it is negative for the reaction. So see, this is a pathway. So this is an energy pathway done by each and every microorganisms as is the living cell. So the TCA cycle and then the EMB cycle. Okay, so certain organisms they will be converting the molecule into acetyl-methylcarbinol. So this acetyl-methylcarbinol can be detected by the test called VP test, that is August-Proskus test. So this is the test, positive test for the VP reaction. So this is the eosin pink color formed at the top of the medium. This is negative test for the August-Proskus reaction. So the next test we are going to see is the citrate utilization test. Okay. So citrate is a very complex compound, so which cannot be utilized by all the bacteria. So certain bacteria they can be utilizing the citrate compound. So in the citrate medium, it will be available as sodium citrate, not in the form of simple citrate. They will be available in the form of sodium citrate. So in that medium, the indicator, which is said to be the bromothymol blue, that has been added already in the medium. Okay, so by the conversion or by the usage of citrate, so the citrate will be decomposed or it can be utilized by the enzyme called citratase. So this citratase has been synthesized by the organism, so that they will be breaking the sodium citrate into oxaloacetate as well as the acetate. So the oxaloacetate and acetate will be splitted out. After that broken down process, the pyruvate will be produced in the medium. So that can be detected by the conversion of the color from green to blue color. So the green color of the medium will be turned into blue color in presence of the pyruvate as well as the carbon dioxide, so that what has been produced in the medium is nothing but the sodium bicarbonate. So this sodium bicarbonate and ammonia, they changes the pH from alkaline. So that alkaline pH changes the indicator bromothymol blue from green to blue color. So this is the positive reaction for the citrate utilization. Okay, so initially the medium will be green in color. Okay, so after the utilization of sodium citrate, this green color will be turned into blue color. So this is the positive reaction and this is the negative reaction. So see, the sodium citrate plus ammonium salt that will be present in the initial medium. When the medium Okay, so when the medium has been utilized by the bacterium with the help of the enzyme citratase, so they converts the medium into sodium bicarbonate plus ammonia with the liberation of alkali pH. So this pH changes the medium from green to blue. So this is the positive reaction. So the next test is the urea utilization test. So this is also recommended for certain kind of Enterobacteriaceae family. So especially the protease species. Okay, so this protease is positive for the urease test. Some other Enterobacteriaceae family are negative for the urea utilization test. So to detect the presence of protease enzyme as well as the protease species, we will be doing this urease utilization test. Okay, so here the medium used has been the Christensen urea agar. Okay, so that enteric bacteria, the enteric bacilli, they have the ability to utilize the urea. So they have an ability to synthesize an enzyme called urease. So that enzyme urease will be splitting up. It will be converting the urea into the negative products. Okay, so their products, and then as a result what happens means alkalinity will be formed in the medium. So they will be converting the pH. Okay, so what is the reagent present in this urease medium means phenol red. So this phenol red will be converting the medium from orange color to the pink color. So this pink color is a positive reaction for the urease utilization test. See. So see, this is the medium. Okay, so this is the negative test. This one is the positive test. So if the organism synthesizes urease enzyme means they will be utilizing the urea present in the medium and converts the medium into the pink colored product. So this is the positive form and this one is the negative test. So next test is the sugar utilization test. So we know that all the organisms have the ability to utilize carbohydrates for their metabolic activities. So in that carbohydrate thing, glucose is the common sugar which has been utilized by all the organisms because it is a simple sugar, so that all organisms have the ability to synthesize and metabolize and ferment, utilize glucose. So other than that, different varieties of sugars will be available in the sugar fermentation process. So in case of Enterobacteriaceae family, all the members of Enterobacteriaceae family, they have an ability of utilizing the glucose. So we need a pattern of sugar fermentation. So other than glucose, what are all the sugars they can able to ferment? So that can be listed up. So likewise, maltose. So maltose fermentation is the best method in which we can able to differentiate the Proteus vulgaris species from Proteus mirabilis species. So in case of Proteus genus, two species can be differentiated using the sugar fermentation test. Proteus vulgaris as well as Proteus mirabilis. So Proteus vulgaris is positive for maltose fermentation and Proteus mirabilis is negative for the maltose fermentation. So what is the common medium or common indicator used for the sugar fermentation test? So in this sugar fermentation test, what is the medium we can use is the sugars, nothing but the sugars. So the common sugars we can be used for the sugar fermentation test. So it will be 5% or 10% depends upon the medium we are selecting. So 5% sugar or 10% sugar. With that we will be adding a pH indicator, that is the phenol red. So this phenol red can be used as a pH indicator so that they can able to convert the acid or alkalic productions in the medium. So when the sugar has been utilized by the organism means definitely by the fermentation mechanism or by the metabolic activities, the acids and alcohols will be liberated in the medium. So that will be converting the medium's pH. So the acid pH or alkalic pH, that can be easily detected by the conversion of the indicator molecules, that phenol red. So that phenol red definitely changes the pH of the medium. So that organic acids produced due to the production of carbohydrate utilization will be easily detected by the phenol red indicators. Other than phenol red indicators, we can use bromocresol purple, that can be used for the detection of sugar fermentation. So see, this is the phenol red indicator added medium. So the medium will be orange in color. Okay, so it will be orange in color. So we have been used 3 different organisms: E. coli, Shigella soni and then Pseudomonas aeruginosa. See, these two organisms, E. coli and Shigella soni, is positive for the sugar fermentation. So what is the sugar that you are used is the, see, so this is the glucose. So the sugar used in this medium is the glucose. Okay, so first thing is the uninoculated tube. So this is the control medium, and the next 3 tubes are the inoculated medium. So first thing is E. coli, second thing is Shigella, and third one is the Pseudomonas. So Pseudomonas is negative for the glucose fermentation. E. coli and so on. So see, in E. coli there will be a gap formation, that will be a bubble formation. So this is to show that it is a gas production. So the medium contains gas. So the empty gap produced in the Durham's tube is nothing but the gas production. This soni, they don't produce the gas, so the color of the medium alone changed into yellow color, but no gas production or bubble formation will be same in the medium. So this is positive reaction with gas production, and this is only a positive reaction due to the acid formation. And this is completely the negative reaction, so we can able to compare the slides by the tubes, which is the control tube and then the culture tube. So this is bromocresol purple. So in the previous slide we have been seeing that phenol red has been used in the medium. Here bromocresol purple, that can be used as an indicator in the medium. But the same reaction will be formed. So when the acid has been produced means the medium will definitely converted into yellow color. There is no change. So same, that gas production will be seen by the empty gas bubble formation in the top of the Durham's tube. So that can be easily detected by the color change. So if the acid has been produced means we can able to see the medium color changes into yellow color. So if there is no production or no utilization means the media will be in the acid is formed. Okay, so this is the positive acid production or the sugar fermentation part. So the next part is the analytical profile index. So this is an advanced form of biochemical detection methods. So in that previous slides we have been seeing different types of biochemicals, but each and every biochemical we have to produce or we have to make it by our own in the manual form. But this is the new advanced form of analytical profile index. So what is that advanced form of profile index means? This is a plastic strip. This analytical profile index is nothing but a plastic strip which has been coated with a different number of biochemicals. Okay, so see. So this is the plastic strip. Okay, so it has been coated with a different number of biochemicals. It may be a sugars or it may be any type of sugar test or it may be any type of metabolic test. So any type of biochemical test, indole, MR, VP, everything can be taken in the profile index. Okay. So what is the principle behind this analytical profile index means? So it composed of a plastic strip with the cupules. So these cupules are nothing but this thing. So we cannot say it as a capsule. So it is a cupule. So why it is a cupule means? So they will be having a hole at the back side. Okay. So through that small opening, we will be inoculating the medium in the thing. As well as after the completion of incubation, we will be adding the reagent through that opening alone. Okay. So that we can able to detect the presence as well as the absence of the reaction. So what is the procedure? A saline suspension of test organism is dropped in the cupules and then the strip is covered with a lid and then placed in the humid plastic chamber. Okay. So the same procedure which has been followed in the manual thing. So while we have been preparing the medium in the test tubes as well as in the form of any other glass vase, same procedure will be used for the inoculation as well as the incubation thing. Same 37 degrees at 24 to 48 hours. After that, the medical, that is the biochemical which has been utilized by the organism. The test suspension we have been added in the cupules, no. So that will be converting the biochemical into positive or negative reaction. So that will be observed with the available standard charts. So each and every analytical profile index, they will be having a standard chart. So see, this is a standard chart. So this is a standard chart. Each and every test, they will be having a substrate. So what is the substrate present in that particular chart? So see, CIT means citrate, URE means urease. Likewise, different number of tests will be available in the analytical profile test. So here the test we are going to detect. So what type of reaction will be done in that cupules? So see, this is tryptophan means that indole production can be done. Okay, so tryptophan in that process, the aminase also can be detected. Likewise, each and every capules will be contained with the tested reactions. The results we have been given. So, if it is negative means they will be producing this type of disease. If it is positive means they will be producing this type of disease, this type of results. Okay. So, with that positive and negative results, we can able to calculate the result. Okay. So, with the help of the positive result, we will be making the chart with the positive and negative signs. So, each and every positive signs they will be having the points. So, that points will be added and then will be converted into digital code. So, that digital code will be compared with the standard chart. So, that finalized digital code will be converted into the organism. So, with the help of digital code, we can able to detect the organism easily. So, this is the advanced form of biochemical test we can able to done in the laboratories. So, that's all about biochemical characterization. Thank you for the cooperation. Thank you.